Okay, so we had done in the previous half that stage 2 thymocytes are now totally ready. They are the double positive cells. Finally, we move on to stage 3 thymocytes. These double positive cells then go under two kind of selection, a positive selection and a negative selection. I'll be dealing with the selections later on. Let me finish this part of the story. So as we see, when we get a double positive cells, so the moment we get a double positive cells, these are stage 2 thymocytes. They are now mature, but the two kinds of schooling needs to be done. The two kinds of selection needs to be done. Now, when these two kinds of selection is completed, the final output we get are the stage 3 thymocytes. They are the mature thymocytes. They have cell surface CD3 associated with alpha beta TCR expressing at a very high density, and they are divided into two distinct subsets. We get two distinct subsets, one the CD4 positive and the other the CD8 positive. Two more important factors to be noted. When the stage 3 thymocytes, which are the mature thymocytes, are finally ready to migrate out of the thymus, they start re-expressing the CD44 receptor. So they had entered with the CD44 receptor. They went through stage 1, stage 2 thymocytes. They became stage 3. Now they re-express the CD44 receptor, which is thought to be involved in migration and homing to the peripheral lymphoid tissues. So they will now move, they will migrate from the thymus to the different lymphoid tissues at the periphery. So for that reason, CD44 receptor is needed. Further, the mature thymocytes or the stage 3 thymocytes also express L-selectin, which is important for adhesion of this lymphocytes if needed to any tissue. So these are the basic 3 stages through which a progenitor lymphocyte enters and a mature thymocyte leaves out. Now let's come into that part where we are going for positive and negative selection. So now let us look into this particular slide. Give me a moment. So as I said, double positive thymocytes, stage 2 thymocytes who have CD8 and CD4 positive cells, both are expressed. And we have the TCR receptor expression also done. There are basically 3 models for positive selection. The output of the positive selection, as we know, is either we get a CD4 positive cell or a CD8 positive cell. At the outset of the class, I had asked all of you to write on your notebook TCR CD4 MHC class 2, TCR CD8 MHC class 1. So TCR CD4 MHC class 2, this TCR CD4 and TCR CD8, so the expression of CD4 on the lymphocyte or the expression of CD8 on the lymphocyte is actually controlled by the MHC receptor to which these co-receptors bind. So it is actually the MHC which is ensuring that whether a cell will have a CD4 receptor or whether a cell will have a CD8 receptor. Hence, this kind of schooling is also known as MHC restricted. So at times it is also said that the lymphocytes are MHC restricted. The terminology means that MHC is restricting the expression of CD4 receptor on helper T cells, and the MHC is restricting the expression of CD8 receptor on the cytotoxic T cells. Now let us see how this actual selection is done. How it is that either the CD4 receptors are activated or either the CD8 receptors are activated. There are three hypothetical models for this particular case. The most accepted being the kinetic signaling model. But before going into that, I'd ask all of you to look into this particular cell. We had discussed this cell in my previous recording. I had said that these are cortical thymic epithelial cells. These cells trap antigen. These are not self antigens. These are non-self antigens. These cells trap non-self antigens, and these cells are capable of expressing both MHC class 1 receptor and MHC class 2 receptor. Since these cells can express both MHC class 1 receptor and MHC class 2 receptor, so they can interact with both CD4 expressing cell and a CD8 expressing cell. Mind it, when it interacts with the, when a developing lymphocyte interacts with the MHC class 1 receptor, it does so by the CD8 molecule. When a developing thymocyte interacts with a class 2 molecule, it interacts via the CD4 receptor. If not, for example, look into this picture. This is MHC class 1 molecule. The green receptors are the MHC class 1 molecule. The red receptors are the MHC class 2 molecule. If I look into the green receptors, and the pink one is the CD8 co-receptor and the blue one is the CD4 co-receptor. So if you go by the color, the blue one should recognize the red one, and the pink one should recognize the green one. If you look into this picture, by mistake, the pink one has recognized the green one. So a CD8 co-receptor has somehow interacted with the MHC class 1. Automatically, this is a negative selection. This is also a form of negative selection. That is, the co-receptor is failing to actually identify its own traditional MHC counterpart. So this is also a form of negative selection. These cells are induced to die by apoptotic death. On the other hand, if the pink one, so the CD8 receptor is recognizing the blue one, the class 1 molecule. So the CD8 is successful in recognizing its cognate MHC receptor. So this is a positive selection. Now, regarding this positive selection, as I said, there are three models. So let me explain the selection process by seeing the models. So as I said, that let's concentrate now on positive selection. How is this selection being done? How is that among the two lymphocyte receptor, co-receptor, one is being selected by a MHC molecule? So let's move into the first model, that is the instructive model. The instructive model says that the interaction of a co-receptor with a molecule, it is a matter of chance. The TCR sits with the MHC class 2 and it is seen that the CD4 is engaging with the MHC class 2. This engagement actually sends a negative feedback signal, as a result of which the CD8 receptor is down-regulated. As you can see, CD4 engagement signal. So we are having a CD4 high expression and a CD8 low, and in course of time, the CD8 gene will be suppressed. On the other hand, this is the CD8 engagement signal. So it's a matter of chance. The lymphocyte is interacting with the TCR, and the CD8 has recognized its cognate receptor MHC class 1, and this is the CD8 engagement signal. So initially we are getting a CD4 low and a CD8 high expression. Mind it, when we have a lymphocyte in this condition, you have to assume that both the genes of CD4 and CD8 are operative. The moment an engagement is occurring, the result of that engagement is inducing a signal transduction pathway, as a result of which the other receptor is gradually being, the other co-receptor is gradually being down-regulated and ultimately suppressed. So this was the instructive model. Who is giving the instruction? The instruction is being given by the MAC molecule, because once the MAC molecule recognizes, if the class 1 recognizes its CD8 co-receptor counterpart, it is instructing that co-receptor to be activated. Automatically the other one is down-regulated. And on the other hand, if the MAC class 2 is recognizing its CD4 co-receptor and activating it, automatically the other one is being down-regulated. So this is the instructive model. The other one is a random model. The random model says that both are highly expressed. Both the cells are being highly expressed. But, I mean to say both the receptors, both the receptors are being highly expressed, but it's a random choice as to which receptor stays. So that is a matter of choice, that as to which receptor is staying. So as you can see, so it is randomly the CD4 expression is upregulated, and automatically the CD8 expression is down-regulated in the second case. So once the CD8 expression is down-regulated, it is unable to bind to, CD4 is high, CD8 is low. So CD8 is not now no more functional. CD4 being high is able to bind with MHC class 2. So it is becoming a CD4 positive cell. On the other hand, just the vice versa. CD4 expression is lowered. It's a random CD4 lowering of expression. CD8 high. It is able to bind to MAC class 1 receptor. CD8 expression is established. The cell is becoming CD8 positive cell. If in this condition, the cell is not being able to bind to a class 1 molecule, or in this condition, if the cell is unable to bind to a class 2 molecule, then automatically apoptosis ensues. So this was the stochastic model. This is a random choice. Why and how, which receptor is chosen first is not been very much clearly described by this model. It is a random selection of any one gene. Once one gene is selected, the other gene automatically gets suppressed. Okay. Now coming to the final and the most accepted model, the kinetic signaling model. This model was proposed by Alfred Singer and his colleagues. And this model is a bit critical model. What did they propose? They proposed that the thymocytes commit to the CD4, I'm again repeating, the thymocytes commit to the CD4 positive T cell lineage if they receive a continuous signal. If they receive a continuous signal in response to TCR co-receptor engagement, but commit to the CD8 lineage if the TCR signal is interrupted. That means the T cell receptor engages with either MHC class. Let us assume that the T cell receptor engages with a MHC class 2 molecule. The moment the T cell receptor engages with the MHC class 2 molecule, a continuous signaling starts between the MHC class 2 molecule and the T cell receptor. As the signal process and the signal strengthens, the CD4 co-receptor expression is heightened. The CD4 receptor gene is established. CD8 receptor expression starts becoming gradually down-regulated. Now, this cell was initially a double positive cell. If it so happens that suddenly in some of the double positive cell, the CD8 co-receptor expression is suddenly becoming down-regulated, these lymphocytes suddenly detach. Not all. For some, the CD4 lineage is established, but for a few, suddenly these lymphocytes detach. The moment they detach, the TCR MHC signaling is disrupted. The moment the TCR MHC signaling is disrupted, they are then revived. They are Singer et al. Further said that these cells where the disruption has occurred, these cells are rescued by the IL-7 So that interleukin 7 now rescues this cell, and these cells develop into the CD8 lineage. That is, their CD8 receptor is further activated, CD4 is down regulated, and they become the CD8 lineage. So this was the disruptive signal hypothesis provided by Singer et al., which showed that if the signal is continuous, then the cells develop into a T helper cell. If somehow the signal is discontinuous, that is, somehow the TCR and the MHC class 2 molecules move apart, then these cells automatically, with the help of the IL-7 microenvironment, are committed to the CD8 positive lineage. Till date, this is the most accepted hypothesis for positive selection. And hence, at the outset of the class, I had asked you to write TCR/CD4/MHC class 2, and I had said hyphen positive selection. So positive selection means automatically all the double positive lymphocytes will engage with the MHC class 2 molecule via their TCR/MHC receptors. And the CD4 expression is stabilized. That is the default pathway. And that is actually positive selection. If a disruption of this signal occurs, then the cells we get are the cytotoxic or the CD8 positive cells. So now we move on to the next part of our selection, the negative selection. So this is a picture of the medulla where the negative selection mainly occurs. And as you can see, these are the medullary epithelial cells. You can see MTC, medullary thymic epithelial cells. Within these epithelial cells, we are seeing this particular name. This is ARIE positive. That means this gene is upregulated. So these cells are now producing this transcription factor, which is also known as autoimmune regulator. So what does this medullary epithelial cell do? These cells tries to express almost all the variety of antigens found in all the tissues of our body as much as possible. So it usually expresses all the self antigens, all the variety of self antigens that are possibly present in our body. It might also at times miss one or two. That happens. That sometimes happens. So as I said, it presents all the self antigens to the already positively selected thymocytes. The thymocytes have completed positive selection. So some have become CD4 positive cells, some have become CD8 positive cells. Now the next part of the schooling, whether the cells are overreactive to self antigens. If the cells are overreactive to self antigens, then there is a risk of sending these antigens to the peripheral immune system. So they need to be killed. So if the cells are seen to be overreactive with self antigens, then as a result of interaction of these overreactive thymocytes with the medullary epithelial cells, they are killed. Only the moderately reactive, that is mild, moderately reactive thymocytes are selected, and the moderately reactive thymocytes are then further proceed to mature as the CD4 positive and the CD8 positive T lymphocytes. When this moderately reactive thymocytes are finally come out of negative selection, they again develop, as I said, the lectin receptor and the CD44 receptor. So these are the two selections that occur, one the positive selection and the other the negative selection for a T lymphocyte to become successfully mature and move into the peripheral lymphatics or to the blood vessels. So this is a very schematic representation. Fourth sem you do not have this in your syllabus, yet I have shown this, that this is how the gene is rearranged. So Astik, you are asking me about the rearrangement of the receptors. So this is a schematic rearrangement of the receptors. That this is the germline DNA. The two genes are rearranged, and this is how the receptor rearrangement is occurring. So this is just a picture. You don't need to study that. This picture is there to have a view of how the genetic rearrangement occurs. These are the models which I've already discussed. Now let me come to last part, because we have already finished what was written, what was there in the syllabus. This is the part where T cell is activated. So once we get proper mature positive CD4 cell or a positive CD8 cell, now the T cells mature. The final maturation, they have matured, sorry. The final activation depends on two signals. A T cell is mature, a T cell is now CD4 positive, and the T cell has now moved into blood. However, these T cells are not always activated. The final activation depends on two factors. One factor is their further interaction with the antigen presenting cell, and the second factor is the production of cytokines from these lymphocytes. So later on in the blood or in the peripheral lymphoid tissues, they further interact with the antigen presenting cells and they produce cytokines. They also upregulate receptors for the cytokines, and thus they further divide, activate, keep a memory population and keep a running activated population in the bloodstream. But this activation usually happens only when the body has an antigenic exposure. If there is no antigenic exposure, usually this kind of activation is not seen. The mature lymphocytes circulate in our blood, but they are not activated unless and until there is an antigenic load in the blood or in the lymphatics. This is a very schematic representation that the CD4 positive population is also known as the helper population or the helper 0 population. And depending on the cytokine microenvironment, the mature C, there we have this IL-12. That means a phagocyte. This is activated by a phagocyte. IL-12 is usually secreted by macrophages, a very classical cytokine. This is IL-4, and here we are having TGF-beta, IL-6, IL-23. So depending on the cytokines produced, whenever there is an antigenic exposure in the blood, the mature CD4 positive lymphocytes or the helper T cells can further differentiate into three basic populations. Each population or each subset has a specific function. Some subset that is being activated by macrophages produce this kind of cytokine, a very specific function. It moves on, as you can see, it activates further macrophages. It helps in antibody switching in case of B lymphocytes. Not only this, it also activates the T cytotoxic cell. So the initial mature CD4 positive T lymphocyte can further be divided into three categories or four categories depending on the microenvironment and the cytokine produced in that microenvironment. These, as you can see in each of the subtypes, you can see these are the master transcription regulators that are then produced in that cytoplasm. That means when a mature CD4 positive T helper cell differentiates into a T helper 1 category, the T beta gene is expressed to produce this transcriptional regulator. And this transcriptional activator or regulator then controls the further functions of these cells. So these are further different. So that was the initial three subtypes. I've given this picture for an overview. This is a mature CD4 T cell. And apart from the above three subtype, one subtype which I did not mention here, you can see is this category. This is a very important category because this cell helps in development of tolerance to T lymphocytes at the periphery. As you saw, the T lymphocytes develops a central tolerance in the medulla by the medullary epithelial cells. But there is also a category of T lymphocytes that its cells are produced from thymocytes and differentiate in the medulla. These lymphocytes are the T regulatory cells. These regulatory cells can also suppress hyperactivated T lymphocytes if it ever occurs in the peripheral region. So as you can see, this is a regulatory T lymphocyte. It can suppress a T helper cell by withdrawing the cytokines it is producing. A T helper cell is producing interleukin-2. So it is suppressing this T helper cell by withdrawing the cytokines, by itself upregulating the receptor. It is itself upregulating its own receptor. It is withdrawing all the cytokines to itself and thus suppressing its further activity. It can produce inhibitory cytokines and can thus suppress the hyperactivated lymphocyte in the periphery. It can interact with MHC class 2. It can interact with MHC class 2 and thus prevent the antigen presenting cell from further antigen presentation. Further, it can induce apoptotic death in a T lymphocyte which has been hyperactivated. So these T regulatory cells play a big role in T cell energy at the periphery. T cell energy or T cell tolerance at the periphery. This is a very schematic representation of the various steps in which a T cell is activated. So APC, antigen presenting cell, first takes up the antigen, then it presents the antigen, then T cell activation is occurring, and after the T cell is activated, if the antigenic load is removed from the blood, then the T cell inactivation is also occurring. So these are the 4 basic steps in which a mature lymphocyte or the thymocyte that comes out into the blood follows a life cycle like this. So, but the life cycle depends on the antigen uptake. So if a mature lymphocyte in its lifetime does not react with any antigen or does not interact with an antigen, the life cycle might not take place in this manner. This is a licensing APC. This is central tolerance. However, fourth sem, this is peripheral tolerance. I think so we do not have much time for this. I'm again sending a link after this one. So I'm pausing the recording and I'm sending a link after I record this slide.